Hi, my name is Rabtah Jinan, and my study is on the comparative conformational and mutational analysis of RNA frameshift elements from RNA viruses. So just as a general overview, during the translation of a genome, the messenger RNA molecule is translated into a sequence of amino acids for protein synthesis. And this process of translation is very important for in the central dogma, which basically converts DNA into proteins to allow protein function for the organism. And an RNA virus is one where the viral RNA can be single-stranded or double-stranded, and the genome can occupy a single RNA segment. And RNA viruses utilize the same process of translation to synthesize proteins and carry out functions for the virus's survival. Now, going into FSE, RNA viruses use the RNA frameshift element, which is FSE, to produce multiple protein molecules from their compact genome and enhance their ability to synthesize proteins. So this mode of protein synthesis regulation is called programmed ribosomal frameshift, or PRF. So the RNA frameshift element is present between two overlapping open reading frames of viral RNA genome, and it consists of a slippery sequence followed by a stretch of nucleotides, and they usually fold upon themselves to attain a specific conformation, which is usually a pseudoknot. So during translation, the ribosome slips through the slippery sequence to encounter the pseudoknot, and then this makes the ribosome backtrack the sequence by a single nucleotide and read the open reading frame in a different manner. So as you can see, just for reference here, when the ribosome moves back by one nucleotide, it starts reading the sequence differently, and this creates different proteins. So this mechanism of PRF is called -1 PRF, and it's essential for the viral replication of the RNA virus and the evasion of the host immune response. So RNA viruses typically mutate more due to the error rate in the RNA reverse transcription, and this also means that RNA viruses have many variants. And variants can escape vaccine immune response because the structure mutates and different functions are carried out, so novel therapeutic interventions are needed to eradicate or contain the whole disease. Now, in terms of our project, because the conformation of the RNA FSE is essential for viral infection and its survival within the host organism, we identified the sequence and folding conformation of the RNA FSE from 4 different RNA viruses. And through mutational analysis, we identified the nucleotides that could be targeted to alter the conformation and their function inside the virus. Overall, our study explores the specific RNA FSE conformation in the virus and how the conformation can be targeted to develop novel anti-therapeutic interventions. Now, going into our methods, the first thing that we did was sequence alignment. So we retrieved the whole genome sequences of the 4 RNA viruses from the NCBI database. So we did hepatitis C virus, the West Nile virus, human immunodeficiency virus, and severe acute respiratory syndrome coronavirus 2. And we used the information from earlier studies to identify the RNA FSE sequence of each virus from the entire genome. Then we submitted the sequences to the sequence alignment program, which is Cluster Omega, to determine the % sequence identity between the FSE from the different RNA viruses. Then we modeled the conformations of the RNA FSEs, and we used IPNOT, which is an RNA modeling algorithm, to model the structures of the viruses. And we fed the RNA nucleotide sequences individually into the web server to predict their conformational landscape. And lastly, in regards to mutational analysis, we selected a string of nucleotides and we mutated them in such a way that they should disrupt the base pairing interactions from each RNA FSE conformation. Then we ran these mutated sequences again through IPNOT to assess any changes in the conformations. Now, going into the results, after performing the sequence alignment using Cluster Omega, we opened the final alignment results here with GelView in this figure. And our results show that FSE is poorly conserved within the various RNA viruses. And the consensus panel, which is the black bar over here, represents the sequence similarity of the RNA FSE between various RNA viruses. So the taller the bar means that the more highly conserved the nucleotides are for that particular position. Then over here, we see the percent identity matrix, and it illustrates that the sequence conservation between the FSE from the various RNA viruses are less than 60%. So this tells us that the RNA virus sequences are not the same. So to understand the conformational landscape of each FSE from the RNA viruses, we ran the IP0 web server. And below are the radial figures of the RNA FSE secondary structures from the different RNA viruses. And these structures illustrate the hydrogen bonding or base pairing interactions between pairs of complementary nucleotide bases. So for example, here in the West Nile virus, we see that the stem 2 of the West Nile virus is folding on the main nucleotide backbone to form a pseudoknotted region, which is stabilized by 7 base pairing interactions. Then for SARS-CoV-2, we see that the apical loop, which is the loop at the end of the stem, the apical loop of stem 4 is connected to the nucleotide backbone, and which is stabilized by the backbone. Then for HIV, we see that the major pseudoknot was formed between the interior bulge of stem 3 and the apical loop of this stem over here. And it's being stabilized by 6 base pairing interactions. And then we see a minor pseudoknot between the apical loop of stem 5 and the nucleotide backbone. Then for hepatitis C virus, we see a very minor pseudoknot domain that's established at the apical loop of stem 4, and it's connected to the internal bulge of stem 3. So to identify the nucleotides that were needed for maintaining the FSE conformation, we mutated the nucleotides that were stabilizing the pseudoknots and in order to eliminate the base pairing interactions. And we reran the mutated sequence through IP0 again. And shown below are the conformations of the wild type, which is the original, and the mutated sequences in the form of arc diagrams that we ran through IP0. The virus sequences are along the bottom of the diagram, so they would be over here. And the arc represents the base pairing interactions within particular nucleotides, and they're labeled as stem 1, stem 2, and stem 3. Regions where the arcs intersect, such as here, represent the pseudoknot domain. So for the West Nile virus, stem 2 intersects with stem 1, thus forming a pseudoknot domain. So I knew to mutate this region over here. And once mutated, the base pairing interactions within stem 2 were disrupted. Then for coronavirus, we see that I mutated this region over here because stem 2, stem 4, and stem 1 intersected. And once I mutated it, it resulted in the disappearance of the pseudoknot between stem 1 and stem 4, and it resulted in a new 3-way stem loop helix between stem 1, stem 2, and stem 3. Then for HIV, stem 3 and stem 4 intersected to form a pseudoknot region, so I mutated this region over here. And instead of breaking the base pairing interactions, the FSE sequence formed a new pseudoknot domain, which is formed by the intersection between stem 1 and stem 3 in this region here. However, it still remains to be seen whether the conformation changes affect the function of the virus. Then for hepatitis C, we see a very weak domain over here between stem 3 and stem 4. And upon mutating it, we were able to completely eliminate the pseudoknot, which left only stem 1, stem 2, and stem 3. So to go into our conclusions and discussions, we identified the RNA FSE sequences from 4 different RNA viruses, which is West Nile virus, hepatitis C, severe acute respiratory syndrome, and human immunodeficiency syndrome, HIV. So we saw that there was a very little sequence conservation between the RNA FSE of these viruses, and the FSEs fold to form very distinct conformations that are unique to the characteristics of each of the RNA viruses. Our sequence alignment and the conformational and mutational analysis of the RNA FSE identified the unique FSE sequence and the conformations that these viruses adopt in order to efficiently carry on their function inside the host organism. So by mutating these certain nucleotides, we were able to successfully alter the FSE conformation for each virus. And in theory, this should effectively interfere with the programmed ribosomal frame shifting inside the virus and contain the viral infection. So in other words, by targeting these conformations, you will disrupt the protein translation mechanism inside the virus and make it a great antiviral target. And thank you to Talaria for giving me this opportunity to conduct this research. I really had a great time. And thank you so much to my mentor, Dr. Abhishek Dey. I honestly couldn't have done this without you. You are really the one that made all of this happen. You're so knowledgeable and you helped me through every step in the way. And I'm so eternally grateful for this. And I have learned so much thanks to you. So thank you, Talaria, for everything.